Welcome. In this class we will discuss on meiosis. Now meiosis is one of the three cell divisions methods that we have discussed. So the first is mitosis, amitosis and finally meiosis. We will be discussing meiosis in detail today. Now what is basically meiosis? In simple terms I can say it is a double division method of a diploid, that's a 2n, and it divides that diploid into 4 haploids, that is 4 independent n identities, each having exactly half the number of chromosomes as the parent cell. Now where do we use meiosis? We already know mitosis we use in somatic cells. Now why do we use meiosis? Meiosis is primarily used in cells which are sexually reproducing. So you have, let's say we talk about human beings, so you have 46 chromosomes or 23 pairs of chromosomes. Now what would happen if that goes on to the second generation? So if we do not reduce this to half, this would double off in the next generation. Therefore, there is a requirement to reduce the pair from both the father and the mother to half, so that ultimately you have zygote formation, and that zygote would again be 2n or diploid. So let's say in human beings you have 23 pairs of chromosomes. Of these 23 pairs, 22 are homologous pairs and only one pair is the sex determining. So you have XY in case of males and XX in case of females. Now this is a diploid chromosome. So you have one pair of chromosome. Now what would happen under meiosis is, this would, first of all, in the first stage of meiosis, we will see the various stages as we move forward. So in the first stage, this would replicate and then it would break. So you would have 4 haploids that would be produced. So you would have either X, X, Y or Y that would be produced. Now these, you have the 4 possible outcomes, and again you would have the 4 possible outcomes here. Finally NN from these would fuse and then you would have the final formation of the zygote. Now this could occur under various ways. So under meiosis we say there are 3 primary methods of producing. So the first is the gametic meiosis or the gametic method. We also call it as a diplontic method. So under diplontic method, what would happen? It's commonly seen in the case of human beings. So you have parents which are 2n or diploid. Now if these diploid fuse together, it would create a much bigger nuclei which won't be feasible. So what happens is, you have n that is produced from these, and these n finally fuse again to form 2n, and this fusion is by means of zygote. So you have meiocytes which are formed, and these meiocytes are either oocytes or spermatocytes based on whether it's a male or a female, and finally you have fusion into zygote which again forms 2n. So you have a kind of diplontic cycle that occurs, or gametic meiosis that occurs in human being. On the other hand, you have haplontic meiosis, and this haplontic meiosis occurs in organisms which are initially haploid. Now when the organism is initially haploid, what would happen? At this zygote formation stage, it would become 2n, but immediately as it becomes 2n, it would undergo meiosis and it would further break into n. So that would again become haploid, but the initial organism here is haploid, and this is commonly seen in the case of fungi and protozoa. The next is diplohaplontic, and this is commonly seen in spore formation, We also call it as the sporic stage. So spores can exist both as N and 2N. So there is an alternate cycle between N and 2N. So you have haploids which produce diploid, undergo meiosis, again produce haploids, and these haploids again undergo fusion and produce a diploid. So that's a kind of alternate N and 2N cycle that goes under diplohaplontic. Now this is the basic idea why we need to understand meiosis. Now why meiosis is important? Let's say if there was no meiosis, what would happen? 46 chromosome from father, 46 chromosome from mother, ultimately 92 chromosome in the child, and this would be the size of nucleus growing on. So there would be the size of nucleus that would enlarge, the genetic material would enlarge, and finally it would lead to the death of organism. So therefore meiosis is important, which reduces the number of chromosomes to half and therefore makes it feasible for the next generation to carry the same number of chromosome as the parent generation has. So that is the basic idea why we understand meiosis. Now the term meiosis was given by Farmer and Moore. Coming back to the cell cycle that we have studied in the first session. Now this cell cycle talks about the first phase, that's the most important phase, is the interphase, and after interphase you have the meiosis phase. Now as we saw in the case of mitosis, we have three phases, G1, S and G2. However, in meiosis you would again have the same phases, but there would be difference. Difference would be the G1 would remain the same, but this S phase would be an enlarged phase, which would be a phase of DNA replication, and again you would have a small G2 phase, or this G2 phase might be absent in case of meiosis. Finally you would have the M phase. Now this meiosis M phase is further divided into M1 phase and M2 phase, that is meiosis 1 and meiosis 2. Both these meiosis would independently have their two processes the same as mitosis, that is the chirogenesis and the cytogenesis. So under both meiosis 1 and meiosis 2 you would have chirogenesis and cytogenesis. Chirogenesis will deal with the breaking of the nucleus, cytogenesis would deal with separation of the cytoplasm. This would first happen in meiosis 1, where there would be reduction in the number of chromosomes, and then this would happen again in meiosis 2. Now meiosis 1 is predominantly, as you can see here, you would have the nucleus that would enlarge, and finally under the meiosis stage it would break apart, and then you would have reduction of the chromosome into half. Now this meiosis 1 is the phase which is known as heterotypic. Meiosis 2 is known as homotypic. Since it is homotypic, it is equational. So you have the same number of chromosomes that are maintained. However, under meiosis 1, it is reductional in nature. So the chromosome number is immediately half under meiosis 1. Now let's understand the two phases of meiosis 1 and meiosis 2. Before that, one more important point, between meiosis 1 and meiosis 2, you have an intermediate phase which is known as interkinesis. And we will see this after we study meiosis 1. Now let's move on to meiosis 1. Under meiosis 1, you would have four phases, the four stages, that is the prophase 1, metaphase 1, anaphase 1 and telophase 1. Now this telophase and cytokinesis in case of meiosis 1 would be more or less together or would occur at the same time. In certain cases where telophase 1 does not occur, prophase 2 would also not occur. So you would have meiosis cases where you might have anaphase 1 directly moving on to metaphase 2. Now in this case, the most important part is the prophase 1. That's the most elaborate of the whole meiosis, and if you are clear with prophase 1, you are done with meiosis. Now let's start with prophase 1. Prophase 1 is followed with 6 stages. The first is the prolaptoteme, which is usually not discussed. Under this stage, you have very thin chromosomes, and most of the chromosomes are not distinguishable. You cannot identify separate chromosomes. The next to proleptotene is the stage which is known as leptotene. So you have the order that goes in this way. So you have the next stage which is leptotene. Under leptotene stage, which is also known as leptonema, you have the nucleus that enlarges. You also call this as a bouquet stage, where you have one point that occurs towards the end and you have the spread out towards the periphery. So you have the centromere that connects at one point and you have a spread out towards the periphery. Again, under animal cell, you would have the centrioles that would be seen with the esterase formation. So that is the basic phase of leptotene. The next phase, which is the zygotene, is important. Under zygotene, what happens is the actual crossing over takes place and you have formation of the synaptonymal complex. Now this synaptonymal complex has one center and two elongated complex that is part of the synaptonymal complex. Again, under zygotene phase, you have two homologous chromosomes which are attached together at a synapse, and then you have the crossing over that takes place. Now crossing over is predominant feature of pecitine. Under pecitine stage, what happens is you have a bivalent that is formed, or you have a tetrad formation that would occur when you have the replication. Now let's say I have two colors here to help you understand the pecitine stage. So that's my first chromosome and that's my second chromosome. Now what would happen under the stage of pecitine and how does crossing over takes place? That's really interesting to understand. So you have the two chromosomes that are found here. Now under pecitine you would have the endonuclease that would act and that would create a breaking point for the chromosomes here. So what would happen is you would have the top chromosome that would break apart, and again on the lower side you would have another chromosome that would break apart. So that was under the action of endonuclease. Now under the action of U protein you would have the nicking off that would occur and there would be gaps that would be formed. Under endonuclease phase you also have the nicking that occurs. So under the action of U protein, what would happen is this would be the top protein and you would have the formation that would be something like this. Now what would happen? Ultimately at this stage you would have the crossing over that would occur and that would be in the presence of R protein. So this would happen as, and you would have, so you have ultimate crossing over here. Now when these chromosomes would separate, what would happen? The top chromosome would have the sections of lower chromosome and the lower chromosome will have sections of the upper chromosome in simple terms if I want to explain. If in simple terms I want to explain. So this would be the resultant phenomena. So what would happen here is you would have elements of this black that would go into blue and this elements of blue that would come into black. So ultimately that is the way how crossing over takes place and the action of the various enzymes. So first you have the endonuclease that acts, then you have the action of U protein, and finally in presence of R protein you have recombination that occurs and we also call it recombinase, and finally you have the crossing over that takes place under pecitine. So that's the basic stage of pecitine. Now the next stage is the diplotene. Under diplotene you have the nucleoprotein complex that finally is dissolved. There is disintegration of the nuclear membrane and the nucleoprotein complex that takes place. The chromosome tries to condense. We also call this as a leptotene chromosome or the Lamb-Rush chromosome stage. The next to it is the dikinesis. Under dikinesis you have the final separation that occurs and you have moving apart of the chromosomes that takes place. So both the bivalent chromosomes try to move apart at this stage and you have the terminalization that takes place. So here a correct word to use would be terminalization, where you mean that you are trying to move apart. So both the chromosomes are trying to move apart. So that's the next phase of the prophase 1, that is the dikinesis. Now the next stages, so prophase 1 already talked about. Now metaphase 1, anaphase 1 and telophase 1 would be similar to what we talked about in mitosis. So under meiosis again you would have arrangement of the chromosomes that occurs at the equatorial plane. However, these chromosomes which are arranged here have a kind of crossing over, so there has already been the exchange of genetic information that has taken place. Towards the Anaphase, they would start to drift apart. So you have the homologous chromosomes which are exchanging the material by crossing over, and now they are starting to drift apart. So there is distribution which is random, that the distribution of the genetic material is not uniform, and finally under telophase and cytokinesis you have separation into two cells. Now this was the stage of meiosis 1. What is the significance of meiosis 1? The first and the foremost important significance is you try to separate the homologous chromosomes. So now there is no more homologous chromosome, we are trying to separate the existing homologous chromosomes. That's the first thing. So separation of homologous chromosome. The next is reduction into half the number of the chromosomes that are present. So the existing number, you have half the number of chromosomes that are present under the telophase 1 stage. Finally, the next important thing is crossing over occurs, and this crossing over occurs by means of synapse and exchange of information as we discussed. So you have crossing over. Again, the genetic material is exchanged, but that exchange is totally random in nature. So you have random distribution of genetic material that occurs under meiosis 1. So it's not exactly the same. You cannot say the two daughter cells would be identical. Neither you can say those two daughter cells would be identical to the parents. So they are neither identical within themselves nor they are identical to the parent cell. So that's the random distribution. Finally, it induces to form the gametes. So you have a diploid moving on to the formation of haploid. That is again important. So moving on from 2n to n, that is from a diploid to a haploid formation would occur, and at this stage you have mutations that can occur. So that is the first stage, that is meiosis 1. Now followed by meiosis 1 is the stage, as we talked about, is the intermediate stage, and that stage is known as interkinesis. During this stage you have protein and RNA synthesis that occur. There is no DNA replication at this stage, and centromeres do replicate at the stage of interkinesis. Next to interkinesis comes the meiosis 2. Now this meiosis 2 is similar to what we have discussed under mitosis, the PPMAT, the same example that we have talked about in the previous lesson. However, there is the difference between the meiosis 2 and meiosis. What is the basic difference? That's the key of this section. So the basic difference here is meiosis 2 always occur in haploid cells. So meiosis 2, a primary requirement is it should be a haploid cell. It cannot occur in a diploid cell. So first of all, it cannot occur in a somatic human cell where you have a diploid configuration. So it must occur in a haploid cell. The next important condition is it should not be preceded by DNA replication. So it's not preceded by DNA replication. The third important characteristics is the two chromatids that are used are dissimilar. So the two chromatids here are dissimilar, they are not identical. And the fourth again important contribution that we talked about is the two daughter cells that are formed, they are not identical among themselves, neither are they identical to the parent cell. So the daughter cells are not identical among themselves nor they are identical to the parent cell. That means there is a total random distribution of genetic material that occurs. So that's again the key point. So these four are the key aspects what differentiates meiosis 2 from mitosis. Now coming on to the various stages of meiosis 2. Under meiosis 2, as we said, if there is no telophase 1, there would be no prophase 2. So if there is telophase 1, there would be prophase 2, and this prophase 2 is normal. You have a kind of nuclear membrane degenerates, you have the centrioles that move apart here, and you have the nuclear division that takes place with chromosomes which are divergent can be seen here. Now what happens under telophase 2 is more or less the reverse of prophase 2, because under telophase you have the nuclear membrane that would regenerate, you have the endoplasmic reticulum and Golgi apparatus that would try to come back. So those are the things that could be seen under telophase 2. Again you have the centriole movements that could be seen here in case of animal cells, you have estral formation, however it would be anestral again in case of plant cell. So that's again important thing. The next is metaphase 2. Under metaphase 2 you have the equatorial arrangement of the chromosomes that could be seen here, and the arrangement is again known as achromatic polar arrangement. So you have the arrangement that could be seen along the equator, and you have a kind of spindle formation which divides the nuclei while arranging it to the equator. The next is the Anaphase 2. So you have the two chromatids that separate apart and move towards the peripheral locations, and finally under telophase you have 4 haploids that are formed. So 1, 2, 3 and 4 haploids that are formed from the 2 daughter cells that were divided under meiosis 1. So these 2 came from meiosis 1, and from these 2 you have the 4 haploid cells that are finally produced under telophase 2. Now after telophase 2 you have the cytokinesis that would occur. So again you would have the cytoplasm formation that would again remain the same. So it would be either by cleavage method in the case of animal cell, or by the method of cell plate formation as in the case of plant cells. So you have fragmentoplasms that would be formed in the plant cells. So that would be again the process of cytokinesis, which would be similar to mitosis. Now what is the difference under cytokinesis in meiosis 2? In meiosis 2 there can be 2 types of cytokinesis. One could be successive and other could be simultaneous. So these are the 2 methods of cytokinesis or division of cytoplasm that could be seen under meiosis 2. So when I say it is simultaneous, that means all the cytoplasmic activity formation takes place towards the end of both the stages. Under successive, it takes place one after the other. So you have completion of one stage, cytoplasm formation, completion of other stage, cytoplasm formation. So that's successive, one after the other. However, under simultaneous, it would be all at once. So all the processes done, and finally you have the cytoplasm formation. So these are the 2 types of cytokinesis that could occur only under meiosis. This cannot happen under mitosis. So that's again important to note. And finally, we'll move on to the significance of meiosis. So meiosis, as we said, is important for gamete formation. It's important for transmission of genetic information from parent cell to the daughter cells. It helps to maintain the chromosome number. Since you are doing a double division, you are moving from a diploid to haploid, that haploid again fuses to form a diploid. You are trying to maintain the number of chromosomes. So that could again occur by 3 methods, as we said, the diplontic, haplontic and the diplohaplontic. Again there is assortment of the chromosomes that would occur, crossing over that would occur, which would lead to exchange of information. At the stage of meiosis 1, there are heavy chances or bright chances of mutations to take place, that would be the changes in the genetic composition, and finally it helps to establish a basic relationship of the organism. So what we have discussed in today's class is the process of meiosis. We will be taking a separate lecture where we will be talking about the differences between mitosis and meiosis, because that's an important topic and we will be considering that in a separate session. If you have any doubts or feedbacks, you can leave those as comment below the video. Have a good day ahead.